Hello, my name is Jeff Hozek. I'm a research scientist at CSIRO in Australia. I'll be talking about probabilistic risk assessment for synthetic genetic vector control using Bayesian analysis. So the World Health Organization has expressed support for the investigation of genetic control strategies for malaria vectors. The World Health Organization has recognized that morbidity targets will not be met in 2020 for malaria, and targets in the near future are at risk as well, and with concerns about the increased cost of intervention and the potential for insecticide resistance. Self-sustaining gene drive as a genetic control strategy may provide durable and affordable control. But in any case, any genetic control strategy using genetically modified mosquitoes should follow a phased development pathway with risk assessments at each stage, as recommended by WHO. Target Malaria is a not-for-profit consortium with partners in Africa, North America, and Europe. Target Malaria is developing a stepwise genetic control strategy in sub-Saharan Africa, beginning with dominant sterile male, followed by paternal male bias, and then synthetic gene drive. CSIRO Data 61 Ecological and Environmental Risk Assessment Team, which I'm a member, has conducted 2 independent risk assessments for dominant sterile male. The first independent risk assessment focused on the accidental release of 10,000 dominant sterile male mosquitoes from contained facilities in Burkina Faso, and the second focused on the field release of 5,000 male DSM mosquitoes. And both risk assessments are available on the web at this link here. So risk assessments for genetically modified organisms typically use qualitative methods, and this table just summarizes the risk approaches used by various organizations internationally. But that's not what is recommended for gene drives necessarily. So the Australian Academy of Sciences recommends including both ecological and evolutionary modeling, ideally within a quantitative assessment. And the National Academy of Sciences, Engineering and Medicine has recommended estimating the probability that gene drive affects endpoints, and also emphasize the incorporation of empirical data within the phased testing pathways with predefined go and no-go decisions. So the genetically modified organism without gene drive, one copy generally has a 50% chance being passed on from a parent to the offspring. So after a few generations, you'd expect a relatively low fraction of the individuals to be carrying the altered gene after introduction. With the gene drive, the altered gene can convert the gene inherited from the other parent, resulting in a more than 50% chance of passing that gene on from the parent to the offspring. And in this case, the altered gene is almost always inherited, and so there's a much higher fraction that after a few generations that would be expected to be carrying the altered gene. So it's this rate of spread that has this heightened focus for quantitative assessments, and in particular, using probability to address uncertainty of magnitudes and rates with respect to gene drive. Our preferred philosophy follows a scientific method, and it's based on using probability to express our beliefs and our understanding about uncertain states of the world. So if we begin with a knowledge base that is formed from experiments and previous observations, but also scientific theory and modeling, we can use that knowledge base to develop a hypothesis or a prediction, a probabilistic prediction that accounts for uncertainty, and that is called the prior. And then we can bring in new data obtained from experiments or observational studies, monitoring programs, surveillance. We can assimilate that data through the likelihood, and that produces an updated prediction, or the posterior. And this posterior contains information from both the original knowledge base and also this new data that came in from this new empirical information that was introduced. And we can use this updated precision to again update our knowledge base. And so we have this cycle, this scientific method where we can use this knowledge base to develop improved predictions and new experiments to test those predictions. And so here we're using Bayes' theorem to capture uncertainty within this process using probability. Risk has one added feature, which is the idea of loss, which assigns value to states of the world that we care about, which may change depending on the decisions that we may take. So the state of the world may be uncertain, and this uncertainty is quantified through Bayes' theorem. And we integrate over this uncertainty and calculate the expected value of loss, and that's what we define as risk. So you might have heard of risk as being defined as likelihood times consequence. So in certain cases, that is the expectation. And so that's the definition we use here. Risk is the expected value of loss. For dominant sterile male risk assessment, there were several community concerns. And here today, I'll just focus on only one. So this, the top row. Will dominant sterile male mosquitoes have a better capacity to transmit pathogens? And the dominant sterile male construct is only expressed in male mosquitoes. And of course, it's female mosquitoes that can bite humans. But the female mosquitoes can carry the construct, but not express it. And so they would be a genetically modified vector. And so a potential endpoint that would address this community concern of a better capacity to transmit pathogens could be the probability of enhanced basic reproduction number as a measure for disease transmission. So if we look at the basic reproduction number, it's composed of several key entomological parameters, such as the human feeding rate, transmission efficiencies from mosquito to human and human to mosquito, the extrinsic incubation period, and the daily mortality rate, or the probability of daily survival. There is also the human recovery rate, such as the recovery from malaria in humans, and also the vector-host ratio. So for this risk assessment, we focused on a relative risk index, which looked at the ratio of the basic reproduction number for genetically engineered strain, so a female vector carrying the dominant sterile male construct. So the basic reproduction number for this GE strain in the numerator, and then the denominator would look at the basic reproduction number for wild type. And if we look at that ratio, then we can see that terms such as the recovery rate and the vector host ratio cancel. Nevertheless, there are still many terms to consider that may depend on whether the vector is wild type or genetically engineered. So we obtained subjective probability distributions, which were listed from scientific experts for these parameters, and we asked how those, for the experts to predict how they would think the mortality rate would vary depending on whether the vector carried the dominant sterile male construct or not. We also had access to empirical data collected from a mark release recapture experiment conducted in Burkina Faso in Bana village. And this experiment was focusing on wild type Anopheles coluzi, and in particular males, but there was a MRR experiment with females as well, which we were able to incorporate to address mortality rates. And this study is used Bayesian inference for an abundance estimate. And at the end of the talk, I'll provide the citations if you're interested in following up from the references given here. And here we're looking at in the yellow dots are the compounds within Bana village, and also the location of recaptures for male MRRs. So for females, we're looking at the mortality rates, and we asked experts to condition their assessments on a large geographic area and to integrate over seasonal influences, to think over the terms of a year. And we included all the experts in an equally weighted Bayesian model average shown by this black line here. So this is a mixture density. And here, which represents all the expert opinions taken to one model. And you can see there's variation around this black line here, given by that Bayesian model average. So for example, the green lines here, there are two experts who are predicting higher chances of high mortality rates, where these green curves are above the black line here. Now, the mark release recapture experiment was conducted at the end of the wet season, and the results from the MRR suggested a slightly higher mortality rate than what the experts were anticipating. So when you condition on this particular data set, it drags the and the posterior on the right-hand side. You can see that the probability distributions are dragged rightwards to higher mortality rates. In addition, the experts that were predicting initially higher mortality rates have been upweighted, and they have a greater weighting in this Bayesian model average shown by this black line. Overall, experts are still represented in this mixture density. So if we look at this ratio of basic reproduction numbers, and we take the log here on the x-axis, and here we're looking at the relative risk index for malaria or Plasmodium falciparum. And we have the dashed line here is the prior, and the solid line is the posterior result, which conditions on the MRR data. And the probability of increased risk is the area under the curves that are to the right of this vertical dashed line. And it turns out that if we condition on the MRR data, the probability under doesn't change that much. So the probability of increased risk for the genetically engineered vector does not change that much. So the area under the curves to the right of this vertical dashed line. But you can see that the solid line does show that there's an increase in uncertainty. That's more spread out. And that's a feature of, as I said, the empirical data was suggesting a slightly higher mortality rate than what the experts had anticipated, and it was shifting their distributions and spreading it out. And you can see that that causes this relative risk index to also have increased uncertainty. And a similar story also holds when we look at another pathogen, unknown virus, ONNV, which is an alpha virus vectored by Anopheles. So the Mark Release Recapture experiment was focusing on male dominant sterile males, actually on wild type male release. And so we could incorporate that data within the Bayesian model and a partial differential equation model for spread. And that's the results shown here. We're looking at the predicted posterior, and in particular, the area of highest density is shown here by this red circle. And at day 2, the highest density is localized around the points of the release. And then due to mortality, that area of highest density, areal density, gradually decreases to as we proceed to day 5, 9 days after release, and finally 12 days after release. So we can use the MRR data for looking at spread and release and trying to estimate abundance as well. And when we talk about disease transmission, we don't necessarily have to eradicate a vector to reduce disease. We could suppress the vector population and decrease the basic reproduction number to the point that transmission can ultimately be decreased or halted or stopped. So just wanted to provide an example here of a Bayesian analysis that was done using prior information within a process model. It was then updated with analogical modeling data, abundance data, and then investigating control strategies. And so there are a range of control strategies here that were investigated and also explored looking at the interaction when they're deployed in concert, which could be the case as well for genetic vector control. It doesn't necessarily have to be deployed independently, but it could be used in concert with other vector control techniques. And so here we can see that there's the observations are shown in red points here for 8 villages. And there's precipitation forcing data shown by the blue line. And we have the model that's fitted to these observations and the prediction expressed around the population abundance within this Bayesian model. And conditioning on these observations allows for improved predictions and learning for the parameters and a better risk assessment that would allow to investigate how the vector population would respond to different intervention strategies. So in this example, the data were collected from the Garki project, which is several decades old from Nigeria. When we look at the range of dominant malaria vectors, of course, the range is very broad. And so we were interested in expanding the amount of publicly available abundance data that would help support investigations for vector control strategies. not just genetic vector control strategy, but also vector control strategies more broadly, because that abundance question is very important. And ideally, we would like to have risk assessments that were fit for purpose, well suited to the geographic regions in which the intervention strategies are being envisioned for deployment. So that would be the ultimate goal. And having a public database would allow exploration of different types of modeling and exploration of different types of strategies that may have varying impact depending on where they're deployed. So we also, and apart from this large spatial problem that we have with very sparse public data, publicly available data, we also have a temporal problem that could be addressed using longitudinal monitoring data. So monitoring data is particularly important because it allows us to capture seasonal cycles, which are very important for population dynamics, and would have an important would it be an important consideration for any model that's anticipating how the population might respond to sustained intervention strategies. So we're very excited for this meeting and happy to participate in the with PAMCA and VectorBase, and like to acknowledge this collaboration with VectorBase, which is very important to us as we collect data not just for this risk assessment, but hopefully for others as well. And we'd like to acknowledge funding from the Foundation for the National Institutes of Health, which has made this project and research possible. And finally, I'd like to conclude by showing the references for the citations given in the presentation. Thank you.